Yeah, welcome everybody. Thanks a lot for joining this wonderful session where we will be talking much more about biomolecular dynamics. We aim to raise awareness about science education. STEM means for science, technology, engineering, and mathematics. So this name STEM is that for, and we started this STEMgrid in early 2021 and began interview and talk series. And today we are delighted to have Dr. Guillaume Bouvignies, and thanks a lot for joining us today. And now I request Professor Rasmus Linser to introduce our speaker. Thanks a lot.
Okay, perfect. Thanks so much, Himanshu. So yeah, I don't have much, but I would just convey to everyone watching that first of all, I'm also very happy to have Guillaume over, and I just want to say a few words about what he's been doing in the past. And so Guillaume Bouvignies did his PhD in the Martin Blackledge lab in Grenoble, so that was from 2004 to 2008. And he was back then concerned with developing NMR methodology, particularly with the aspect of a simultaneous characterization of the structure of those proteins as well as their dynamics. And then consequently, he moved on and did his postdoc in the Lewis K lab in Toronto. So that was then from 2008 to 2013. And he was very successful there in characterizing and also really developing a lot of methodology for these transiently formed species, invisible states, and computation of their properties, populations, structural characteristics, and so on. And so that has been very path leading for the community, and also for himself, as from then on his scientific way led him to Paris, and he's since been a CNRS scientist and interested in all sorts of novel NMR techniques for studying biomolecular structure and dynamics, and then obviously to use and apply those techniques to gain insight regarding the role of conformational dynamics in proteins of all kinds of applications. And so his interests cover the whole range, starting from this methodological part, theoretical, physical part of NMR, regarding pulse sequence design and data analysis, molecular modeling and the programming of dealing with these data, up to the application side and what to exploit these data and these accesses to. Good, and so with this, I would just like to thank you again, Guillaume, and yeah, the virtual floor is yours.
Okay, but thank you for the very kind invitation, Himanshu and Rasmus, for the nice introduction. And yeah, I feel very honored for your interest in ChemEx. So I'm going to start sharing my screen. Hopefully that should work. If everything is working, you should see my slides now.
Yes,
okay.
It's working.
Perfect. So yeah, today I'm going to talk about ChemEx. So that's a program I developed. It's an open source Python application that is designed to analyze NMR chemical exchange data, and mostly in the context of studying protein dynamics and more specifically characterizing sparsely populated states of protein. So what I'm going to do is probably divide my talk in mostly three parts. The first part where I would discuss or give some background on protein dynamics and the use of NMR to characterize sparsely populated states. Then I would move on to describe the philosophy behind ChemEx, how it works, why I chose to make it work this way, and then a live demo of a few examples, which hopefully will give you enough detail for you to run your own calculation with the program. So yeah, I'd like first to begin with a few words about protein dynamics. Actually, I should say biomolecules dynamics, because that would apply also to nucleic acid, for example. And if I can, all right, now it works. And you probably know that biomolecules are far from being rigid systems. Instead, they are highly or inherently dynamic molecules. And if you think about it, it's not so surprising, as they fold via non-covalent interaction that are pretty weak, actually weak enough to be constantly perturbed by the thermal fluctuations. So if you see in terms of energy, it's more or less the same order of magnitudes between the thermal fluctuation at 25 and many of the non-covalent interactions in water. So it results in a pretty complex energy landscape where the protein or the biomolecules exchanges between multiple states on a broad range of timescale. Here I'm showing, maybe I can do that, okay. I'm showing a very schematic representation of an energy landscape with just one dimension on the x-axis, and we can say, for example, that's the protein structure. And on the y-axis, that would be the associated free energy. And the vast majority of the molecules in the sample would sit at the bottom of that landscape. And this is basically the conformation that would be seen in many biophysical techniques. For example, this is most likely the structure you would obtain when you crystallize your protein. And that's mostly what your NMR spectrum will show to you, the bottom here of that energy landscape. But because of the thermal fluctuation, actually some molecules will populate alternate states, like in local minima, like the one I'm showing here. Those can be with a quite distinct conformation than the one from the bottom of the energy landscape. And different conformation means also maybe different functionalities. So it may be that this is this higher energy states that is sometimes called like a thermally excited states. Maybe you heard about those states with this name. It may be that they are the states that are important for the protein function or sometimes even malfunction. So those states are actually involved in many biological processes, from protein folding, enzyme catalysis, to sometimes aggregation processes. So if we want to really understand the relationship between a protein structure, dynamics, and function, not only do we have to characterize the bottom of the energy landscape, but we also have to include in our study those higher energy states. But the problem here is that those higher energy states are also most often very sparsely populated. It's actually not so surprising, because like if you take a state with an energy that is higher than the ground state by, let's say, a few kilocalories per mole, then the population of that state would be around 1%, no more than that. And 1 or 2 kcal per mole, that's basically the energy of one hydrogen bond. So that's very easily achieved. And then, yeah, the state is very lowly populated. Actually, there's even more like problem that can be even more serious than that for NMR, is that those higher energy states are also short-lived. And if, for example, you take a simple system exchanging between two states where the populations are uneven, so you have an excited state that is lowly populated, it also means that the lifetime of the excited state is much shorter than the lifetime of the ground state. And if that exchange is about the millisecond timescale, or the lifetime is on the millisecond timescale, then the excited states, like basically in NMR, when the lifetime is short, the line width is broad. And in that specific case, the line width would be like so broad that it would melt into the baseline and then won't be visible in the NMR spectra. So just showing a 1D spectrum of one probe, one spin probe in a molecule, exchanging between two states with two different chemical shifts. So that would be the resonance of the major state and the resonance of the excited states. So there you wouldn't see that. So how do you study a state that you can't see in your NMR spectrum? Well, fortunately, there's still some sign of the presence of that state on the major state resonance that also experienced some line broadening. And one series of experiments that have been designed to study those sparsely populated states builds upon like experiments designed like in the 50s. And instead of talking about the spin dynamics directly or spin physics, I would use that analogy with runners and walkers that I've borrowed from Louis Kay. And I think that's a very powerful picture to understand the experiments I'm going to describe. And the analogy is as is. So the major state and the excited state resonate at two different frequencies. So that means that the magnetization processes at two different speeds. And the spin in the major state would be represented with walkers, and the spin in the excited state by runners. And the experiment I'm going to talk about is the spin echo experiment. So it's based on ideas from Erwin Hahn in the 50s. And it's a very simple experiment. So with our runners and walkers, it just consists of a race where we ask the runners and the walkers to go in one direction. And after a time tau, we ask them to turn around and go back the other direction. Now, in an NMR experiment, that would be a refocusing pulse. And it's pretty obvious then that after another time tau, all the walkers and runners, independently on their speed, would cross the starting line at the same time. And that would basically create a spontaneous ordering, and that we would refer to that as an echo. So this is what would happen if all the walkers would keep walking and all the runners would keep running. Now imagine that you have a stochastic process, like you have walkers that feel ashamed that start running, and the runners get tired and start walking, and vice versa. If you do the same rate, then the result is quite different. Here, if a runner or a walker starts running in the middle of the race, you would break the symmetry between the two halves of the experiments. And then at the end of the race, you would have a distribution of runners and walkers. So it's not like all of them would cross the line at the same time. And the Carr-Purcell-Melboul-Gill relaxation dispersion experiments basically consist of doing this race multiple times with different number of refocusing pulses. So we now ask our runners and walkers to change directions more than once. So 2 times, 3 times, 4 times, even more during the same duration. So we keep the duration the same. And it's pretty clear that the more refocusing pulses we add, the less the runners and walkers will be able to get out of phase with each other. And the narrower the distribution at the end of the race will be. Okay, so that relaxation dispersion experiment is basically that, running that experiment multiple times, tracking basically the modulation of that distribution and getting information about the exchange. So if we go back to our spin systems now and we look at the phase evolution of our spins in the transverse plane, here I have multiple trajectories. Basically, each trajectory corresponds to one probe in one molecule, and we have many molecules in our sample, so many trajectories. And because there is the exchange, the trajectories are different from molecules to molecules due to the stochastic effect of it. And we see that it gets out of phase pretty efficiently here with only 2 pulses, but much less when we are pulsing much faster. And the distribution here is much narrower. In our experiments, what we will observe, so it's one of those, the peak corresponding to this spin probe. in multiple experiments, and we see that the intensity of the peaks will basically increase. So the narrower the distribution, the more intense the peak will be, and that intensity can be directly translated to an effective transverse relaxation rate. And by doing so, we can plot the effective relaxation rate as a function of frequency of pulsing, and that's the well-known relaxation dispersion curves that you would observe. So if you have exchange, you have this kind of dispersion. If there's no exchange, that would be a flat line here. You wouldn't observe any difference in any of those experiments. So the CPMG experiments is pretty efficient at detecting minor states, even though they are populated to around 1% or less. You can obtain, when you analyze the profiles, kinetic and thermodynamic information from the rates of the exchange and the populations of the states. And you can also have site-specific information, or mostly structural information, for example, from the chemical shifts of the minor species. Another experiment that became pretty popular for studying protein dynamics over the last 10 years now is the chemical exchange saturation transfer experiments. And this one is even simpler, I think, to describe as a CPMG experiment. It basically consists of a very weak, an experiment when you apply a very weak B1 field at a specific position. So it's weak, therefore it's selective. And you record your experiment and you monitor basically the peak intensity of your major state, the one you see. And instead of running the experiment once, you do it multiple times, scanning in successive experiments each part of the spectrum. So you move the position of the B1 field, and you're searching for that invisible state. So I'm showing it here, but normally you don't see it in your experiment. But one of the experiments will irradiate right at the position of the invisible state. And what will happen here is that you'll start having a mutation of the magnetization in that state. And this perturbation due to the chemical exchange process will be transferred to the major state species, and that translates into a loss of intensity. So what you obtain basically in that experiment is this kind of profiles where the intensity of the major state species is plotted as a function of the position of the irradiation of the B1 field. And you have a modulation of the intensity with a major dip of the intensity at the position of the major state, because you're basically saturating your magnetization, but also a minor dip at the position of the excited state. So although you don't see any resonance in your standard Fourier transform spectrum for this hypothetical system, then you would see it in the CES profile due to the, basically there's some amplification process going on due to the long irradiation. So as for CPMG experiments, you get a detection of very sparsely populated states. You also have the same kind of kinetic and thermodynamic information about the exchange process. You have directly the chemical shift. You don't even already have to analyze your profile. You see it. It's pretty straightforward. But you also have access when you analyze those profiles to the line width of the minor species, which can provide you information about other dynamic processes, basically. So both CPMG and CEST experiments are recorded as pseudo 3D spectra most of the time. So it's a series of 2D planes. Actually, it can be even more than 2D. It can be 3D or 4D planes, whatever, if you have the time. But most of the time, you would record like 2D planes. And in each of those 2D planes, you would vary one of the parameters. So for CEST, it would be the position of the irradiation frequency. And for CPMG, that would be the number of pulses you're applying. And basically what you would do then is to extract the intensity of each of those peaks over the pseudo dimension. And for CEST experiments, this would be basically your CEST profile. And for CPMG, that would be your dispersion profile. And the process of recording those experiments is and analyzing them is pretty straightforward. You have your sample, you collect your data. After processing them, you have a pseudo 2D or pseudo 3D or pseudo 4D, whatever. You extract your intensity. So here, I'll say a few words about that step. You have multiple programs that can help you with that. Most common one is the NMR pipe. There's a autofit.tcl script that does line shape fitting, and so to extract the peak intensities, it's pretty efficient. FUDA by Fleming Hansen is also a very nice option with more line shapes actually than NMR pipe. I also wrote a small script that I've never really advertised, but does basically the same as the two other program with some optimization for specific cases like CEST experiments where you can have a lot of planes, and that can be a limitation for some of those programs. So once you have the intensity profile, so I'm showing a CEST experiment, but that can be also for a CPMG experiment, then you analyze them directly with the program ChemEx. So that's basically what I'm going to talk about in the rest of that talk. And you have here the line of best fit, and of course, next to that, you have all the chemical exchange parameters that are extracted. As I said, for the getting the peak intensity, so here I'm just describing the script I wrote, which is called peak feed, but not really advertised because there's no documentation. It's not something I really thought I would share, but the philosophy of it is really the same as the one in NMR pipe or in FUDA. And basically, the idea is to fit the same line shape over all the pseudo dimensions for each of the resonances, but different intensities. And what you really have to understand here is that the only parameter that changes over the pseudo dimension is really the intensity of the peak. All the indirect dimensions, like the direct dimension and the indirect Fourier transform dimensions, are exactly the same spectrum for spectrum. If to a very good approximation, that's true. Of course, sometimes you are hitting a little bit more in CPMG experiment one plane than the other, but normally there shouldn't be any difference in your spectrum except for the intensity. And the advantage of fitting the line shape is that if you have overlapped peaks or partially overlapped peaks, then you can identify those clusters and fit all the peaks all together so that you deconvolute the contribution of each peak individually. So basically, if there's two peaks that are very close to each other, you may have contaminations if you just take the peak height. But if you take the line shape into account, normally you are separating the contribution of each of the peaks. At the end, and when you run ChemEx, what data you have is a list of intensities. That's the central column here, associated to an uncertainty, and the parameter of the experiment that is varied. So here, for example, that the CEST offset, so the position from the carrier in Hertz. This is a profile that is represented to illustrate that, that text file here. And here is just to show what partially overlapped peak can be, like, for example, in that specific case. You have like one peak that is weaker than the rest of the peak. So it's very likely that that peak number 5 would contribute a fair amount to peak 105. So if you fit the line shape of that peak 5, normally you shouldn't see it in 105 and should be a case. But anyway, you should always be a bit careful when you have this kind of spectrum. So when you have your profiles, you want to analyze them. And there are different ways of doing the analysis. The most standard way for CPMG experiments, at least, has been to use analytical formula linking the experimental data with the exchange parameters. So for example, there's a well-known Carver-Richard equation for the CPMG experiments. There's a great advantage of doing so. In general, it's extremely fast to calculate things this way. The downside of this approach is that all those analytical formula would be most of the time approximative solutions, and in all cases, they are derived from ideal experiments. And all pulse sequences are not exactly that ideal experiments. You have phase cycling, and you have many other aspects that are not perfect. Like, for example, pulse are finite duration. You may have like other relaxation pathways if you have multiple spin systems that are active during the CPMG. Phase cycling can be also different. And for all those reasons, another approach that can be extremely powerful is to basically do numerical integration of the Bloch-McConnell equations. So this approach is very accurate because you can take all the pulse sequence imperfection into account. All the details about the pulse sequence are possible to take into account, so including phase cycling. It's an approach that is actually pretty versatile because you're not, you can simulate any kind of pulse sequence with this approach. Not all the effects, maybe, but maybe like if you have strong couplings or this kind of thing that might be difficult to take into account, not impossible, but take much more time. But almost all the experiments I'm going to talk about can be simulated this way. The downside of the approach is that, yeah, sure, it is actually slower. But with the computers we have now and the library that are now pretty efficient, it's not terribly slower. And so it's really manageable and we can really go that fast. So to explain to you the approach in more details, so I will take a very simple system exchanging between two different states, okay? And one probe with two different chemical shifts between the state A and the state B. So from these systems, it's very easy to derive the kinetic equations. And it's homogeneous first order differential equations with the rates, forward rates and backward rates, can be put in the form of a matrix equation. And if you, well, the most basic equation in NMR, the Bloch equation, will describe the magnetization evolution. If you have a one spin, isolated one spin, and it's basically also a homogeneous first order, no, not homogeneous in that case, but a first order differential equation. And McConnell realized that those two equations are very similar to each other. And it's very easy actually to combine them to have the magnetization trajectory in a system with a chemical exchange in it. And basically what you would obtain is basically the coordinate of the magnetization for state A and state B as a first order differential equation. So here, of course, you have the equilibrium. So it's not homogeneous, but it's actually pretty simple to make it a homogeneous equation. And such equations is very easily solvable. I mean, the solution is pretty straightforward, as this is just the magnetization vector at time 0 multiplied by the propagator. So most of the time we would call this matrix here the Liouvillean. So that's why it's called L here. And when you take the exponential multiplied by the time, then this is a propagator, and that allows you to propagate and get the magnetization trajectory over the time. Okay, so that operation here, this exponential of a matrix, can be done very easily with scientific libraries these days. So numerically. And I've been showing, this is the equation if you have just a single spin exchanging between two states, but of course the system can be more complicated, and the idea remains. You can still get the same type of equations, except that now the magnetization vector can be more complicated. For example, this is for a weakly coupled NH moiety. Here you see all the components of the magnetization. and you can also describe the Liouvillean for each of the states. So it's larger, but it's not difficult to get the different terms in it. You have one of those matrix for each of those states. So it's basically at the end, you have a Liouvillean that is a 30 by 30 size matrix. If you take the equilibrium, it's you have one more row in column, but the idea is exactly the same, and the computer wouldn't make a difference between a single spin or a larger matrix this way. Now, in your calculations, you can do exactly what I've done, but to simulate pulse sequences. So what I've showed is for a static Liouvillean. Pulse sequences, you have static Liouvilleans block by block. So for example, if you want to simulate a spin echo, you have to simulate the first delay. So it's a free precession that corresponds to a free precession Liouvillean. Then you have the 180-degree pulse in the middle. Here, that's the same Liouvillean, except that you're applying a B1 field for duration T pi. So this is a second propagator. And then you have to get the last delay, and that's the same propagator as the first one. So by doing that multiplication, so it's a vector matrix multiplication, you obtain the magnetization at the end of your spin echo. If you want to do it with multiple echo, like this is the case in CPMG experiments, then that's the same calculation. You just have to change the different multiplications. You can take the power directly here in that case, and that would give you the magnetization at the end of your experiments. For a CEST experiment, it's very simple because there's only one block here of irradiation, and that's enough to describe the trajectory of your magnetization. So that's the same matrix as previously showed. There's a B1 field, except in this specific case. You have all the parameters from the exchange here. So the parameters that depends on the spectroscopic parameters like the correlation and the chemical shift and so on. For CEST experiment, there's one more aspect to take into account. It's the B1 homogeneity. It's in general what would dephase very efficiently the magnetization when all the part of the magnetization that's not along the effective field would get dephased pretty efficiently. So this part is actually pretty important for some of the offset in the profile around the major state frequency. So ChemEx would do exactly that for many experiments. So there's a catalog of experiments of pulse sequences that have been implemented. So you can pick the one that corresponds to the pulse sequence you run, but it won't simulate exactly the full pulse sequence. There are parts of the pulse sequences that don't vary, and those parts, they are in general not simulated. The only ones that are simulated are the ones that would vary. So for example, the CEST irradiation here is a part of the pulse sequence that would vary in the pseudo dimension. Or in a CPMG experiment, that would be the number of pulses. So this block here would be the block that would be simulated. Those ones would change the magnetization more or less the same in all the pseudo dimensions, so over the different experiments. So you see, for example, for this CPMG experiment, we are far from the ideal Carver-Richard experiment. We have phase cycling on the 180-degree pulses. We have a central block here that may vary depending on the scan number. So all of that needs to be taken into account if we want to be accurate. Another aspect I haven't mentioned so far, but that's actually very important in the why we are doing it this way, is if you take the CEST experiment or the CPMG experiments, basically the Liouvilleans would depend on the same set of parameters. It means that if you are super accurate in the way you simulate your experiments, then you are able to use the same Liouvillean to simulate two different type of experiments, for example, CEST and CPMG. And then it's very easy to combine different kind of experiments together and use a complementary between, for example, CPMG and CEST experiments. And if you use approximations to simulate the experiments with like analytical formula, then the approximations between CPMG and CEST experiment might not be exactly the same, and it starts to be difficult because you would have bias that would depend on the type of experiments you are trying to fit. So this approach is actually not new at all, and all the formalism has been very nicely described by Peter Allard, Magnus Elstrand, and Torleif Hard in a series of papers. So I'm just showing two references here, and if you're interested in the details, I think they are very nice papers. And ChemEx is really following what's described in those two papers. So just a few bullet points to emphasize some of the specificities here and recall. So ChemEx would use numerical simulation of the pulse sequence. And this way we can simulate evolution during pulses. All non-ideal behaviors can also be taken into account, like off-resonance effects. That's something I haven't mentioned explicitly, but may be important. It, of course, requires, therefore, pulse sequence information. So you would have to provide to ChemEx some details about your experiments, like the pulse width, the carrier position, and of course a peak list to get the information of the chemical shift of the major species. So it's a program that is highly modular. It's been developed at the beginning to be able to fit different kind of experiments together. So right now it's mostly CPMG and CEST experiments. There's also some sign experiments like shift. It's basically when you have chemical exchange, peak position would be affected by the exchange. And this is some information you can extract and get information like complementary information about the exchange. It can be very interesting to get. So the peak shift between an HSQC and HNQC, for example, can be taken in ChemEx and get back to use some information. It's always a work in progress. It's modular, so it means that new experiments can be included, and in the future there will be some that exchange, R1 row experiments, and many, and even new ideas would be easy to implement into the program. If you type ChemEx in Google, if you want to find it, that's what you would get. So there's actually a coffee pot that is called ChemEx, a brand, and that's not the program, but if you type ChemEx NMR, then you will get the GitHub website, and that's where you can get most of the details about the program. In the near future, I think in the coming days, actually, I will just publish a website with the documentation and where the way you should install it and so on is normally well described. That would be a first iteration of the website, but it will get better over time. Pretty sure. Just a few information for you. If you are running into issues or you have ideas for new features, you can go on that GitHub webpage. There are issues page, and I think there will be a discussion page soon where you can even discuss some aspect, or if there's difficulties with the program, that would be the way to go for getting help. All right. So how do you run the program? So right now, ChemEx is a command line application. So that would be nice to have a graphical user interface, but I never really got the time to write it. But hopefully it's not too difficult to run anyway. And if you are familiar with command line application, then that shouldn't be an issue at all. It's a Python application, but that's not important for the user. You don't need to know Python to run ChemEx. Of course, if you want to contribute, then you need to know a little bit of Python. And the thing you really need to get before your analysis with ChemEx is the intensity profiles. So for CPMG or CES, they are the same format, intensity, error, and the parameter that correspond to the intensity, number of cycle in the CPMG experiment, of the offset position in the CES experiment. And then you should be good to go. So from there, I have two options. I can maybe briefly describe to you the different files that are required to run a calculation. I can directly go to the terminal and show during the live demo the different files. But I guess I can maybe do a quick overview of the different files and the way they can get organized. So all examples that I'm going to show are actually available on the GitHub page. There's a directory called examples, and there's one per experiment, and there's also combination of experiments fit together. And on the website that is not ready yet, but will be soon ready. Basically, sorry. Okay, I'll check later, but I have to run the server to get the website. But in the examples, the organization is the one I'm using, but it's not necessarily the one you should use. So in general, you have a folder with your data sets, and the data set will be your intensity profiles. So you can get a folder for each experiment. So it's like 2 CEST experiments here. And inside those 2 folders, you have the intensity profiles, one per peak. You can copy them here, or you can link them. It's up to you. You need an experiment file for each experiment that would describe basically the experiment. So you have the name of the module in ChemEx. Here it's for N15 CEST experiment. The details about the pulse sequence, so the duration of the irradiation, for example, the position of the carrier, the B1 frequency. You have the experimental conditions about the sample. So depending on the kinetic model you're using, so I haven't mentioned that, but you have multiple kinetic models that are available in ChemEx, from 1 to 4 state exchange. So it's available, but you also have like binding models, models that depend on the temperature, or for binding models, you need the protein and ligand concentration, and these kind of things are just put in that part, the condition section. And then there's a section data, where you would put the location of your data sets, so the folder where they are located in, a few additional parameters that can provide information on, for example, if your uncertainty is not really well estimated, there are ways in ChemEx to use duplicates. If you have duplicate points, or the scatter in CEST experiments can be used to estimate the error pretty efficiently. And then you have a subsection, data profiles, where you put all the file names of your profiles and the associated assignment. So this assignment is, if you are familiar with the Sparky peak name convention, it's exactly the one that is used for ChemEx. So if it's a 1-spin experiment, like for CEST, N15 CEST, then the number, the position of the residue and the nucleus associated is basically the only thing you need there. Okay. When you start your fit, it's You're basically extracting the information by least squares optimization. The algorithms that are used, I haven't mentioned them, but they are based on the optimization algorithms that are in SciPy. It's a Python library, and it's wrapped by the library LMFit, which is actually should have put the website, because that's a very handy library to run fits with Python. But you need initial values for nonlinear minimization algorithms. And you have default values given by ChemEx, but you can provide yours. For example, some that can't be defined by default if the chemical shift of the A state. So here in ChemEx, that would be called CS for chemical shift underscore A. And then you give it in ppm. You have some global parameters, so those would be applied if there are many residues. For example, if you put a CS underscore A here and a value here, that would be applied to all the residues. For the population, it does not depend on population, so on the residue number. So it's given in global. And you also have the possibility to define, for example, rotational diffusion correlation time that is used to estimate your excitation rates, R2, R1, or even more complex rates in two-spin systems. If you know the size of your protein, in general, you know more or less an estimate of your tau-c, and then you can use that. It's in nanosecond here. So this file is a parameter file that you have to write in the same format as the others. Each experiment depends on different parameters, so you are not expected to know exactly what are those parameters. But on the website, all the experiments that are implemented in ChemEx would have a page with a sample configuration file where you have all the parameters that can be defined and a description of what they are and in which units they are. You have to provide them. Okay. Another aspect of ChemEx is you can run simple fits, or you can run multi-step fits. And for example, you can select a subset of residues in the first step, fit your population and exchange rate, and do a second step where you fix the population and the exchange rate and fit all the other parameters of all the residues. So you can define that in another file. So I'm calling it method.toml, whatever. And you have the step one include for the residue number that are included, and the step two where you fix the population and the exchange rate, but you fit all the residues. This is also a file where you can define a relationship between parameters. So that's an issue here. What I wanted to write is if you want the R2 of the B state to be equal to the R2 of the A state, you can simply write it this way, except here it should be A instead of B, but otherwise that would just be that. There are also ways of running grid search. Maybe I'm going to show you that later on. You have statistics methods like bootstrap analysis, Monte Carlo analysis for the uncertainty of the things. All of that can be defined in that file. And if you run ChemEx, it's basically a command line. So you have different modules in ChemEx. I'm not going into each of them, but the main one and the one you would be most likely using is the fits module. You have to provide the name of the experiment file. So here it's a star, just selecting all the experiment files in the directory. You can do it this way. The parameter file where you have the starting values of some of the parameters. The method file where you define the different steps. It's optional, you don't really need that. If you don't provide it, then it fits all the residues. And some most, like the commonly fitted parameters are fitted. And you can also give a location for the where files, the output files are written. Okay, and with that, I think I would just move directly to a few examples. And I would like to start with a very simple example where it's a study of a sparsely populated state, but not like of one protein. It's actually a binding reaction. It's a binding reaction between an SH3 domain and a small peptide. And in this system, it was used actually to cross-validate exchange methodology like CPMG or even CEST. It's very nice because you can get the spectrum of the small domain, SH3 domain here free. And also you can saturate the complex and get the spectrum of the complex. But you can also add a little bit of the peptide, like in that specific case, it's about 7%. And you get 7% of the bond state. But what you observe is a free state spectrum. So you can run CPMG experiments to get the information about the bond state, although you don't see it in your spectrum. So with that, I'm just going to move to the example. So here it's a terminal. Commands are, if you're not familiar with the terminal, there are just a few commands to know about. So here I'm just listing a different example I have. So the one I want to show you is in CPMG. Here the different directories I showed to you previously. I can show you the different files. It's exactly the same here. It's a text editor. So the data, we have data collected at 500 megahertz on the 500 megahertz spectrometer. So this is intensity profiles for residue 1, 3, 4, 5, and so on. The same at 800 megahertz. And you have the two experiments file with, for CPMG, you see that you have a pulse width of the 90-degree pulse for simulating the CPMG. The name of the module here is CPMG, the carrier position, and so on, the duration. There are some other aspects like features I'm not going to describe too much, but you can also have a labeling. When you estimate the relaxation rates, if your protein is predaturated, then that's slightly different. You can give that information here, but it's not critical. And the methods I'm going to run is that very simple one I showed you previously, where I'm just selecting a few residue that I know have nice dispersion and fitting the population and the exchange rate. And then in the second part, I fit all the residues, but I'm fixing the population and the exchange rate to the value that were obtained in the first step. Okay, and the command I'm running, so I saved it in a small file just to save you time, so that you don't have to type that all the time. But if I start that, it's just running this file. And that's basically the progression of the fits. You have a lot of information that are printed on the screen. So it starts with you have the version of ChemEx you're using. It's reading the data, the profiles, and the different experiments, reading the methods. Then you have the first step where only a few 10 profiles are taken and it's fitted. You have the chi-square and the reduced chi-square. The output files that I'm going to show you are located in a directory with the name of the step. So I use step 1, but it can be any name. And you also have like plots. And that's most of the time the files that you need to check if you want to see if your fits is correct or not. And I'm going to show you at least maybe 2 of those. Oops. So they are PDF files, so you can open them with any application. So for example, here, this is what you would obtain. So you have the, so although ChemEx is fitting the intensities, when it displays CPMG experiment, it does it the standard way, which means you have the R2 profile as a function of new CPMG. So that's for the different residues, and that's 5 profiles at 800 megahertz, and you have the same profiles normally at 500 megahertz. And here you have the residual. So if you have like that's the distance between the experimental point and the back calculated point. All right, so here you see the fits are good, and you have the same files for step 2. And this is just more residues. But you see those dispersions are flat, most of them. So you think it's noisy, but actually the error bar is pretty small, and it's just because it's flat. So that's why I didn't use them to get the population and the exchange rate in that specific case. What you obtained in the output directory, that's multiple files. So you have the data here, which is the experimental intensity, the error, and the back calculated intensity. You have a parameters directory with the fitted parameter. So you have the fitted value, the estimated uncertainty. This is all the parameters that have been fitted. So you see that there's a chemical shift difference, delta omega AB in ppm for the profiles that were fitted, and R2 at 500 megahertz and R2 at 800 megahertz. And in that specific case, because with our CPMG relaxation dispersion, you can't distinguish R2A from R2B. R2B is constrained. So you have a file of the fixed parameter that were used, so the chemical shift, for example, but they were not fitted, or the R1, which is important because when you apply a 180-degree pulse, if you're off resonance, you would spend some time along Z, and relaxation here is effective with R1, so you have that information here. You also have the parameters that were constrained. So there are some that just are calculated for, because that's nice to have KAB and not the sum of the two. So they are calculated from the exchange rate and the population of the B state, and KBA is exchange rate population. You have the population of the state A, although it's population of state B that was fitted. And the chemical shift for state B, and the R1, because for state B, they were equal to state A, and same for R2. And finally, you have the plot directory where you have the PDF files I showed to you, and the two files that could be used to have the, that were used to make those plots. So if you want to remake those plots, everything is here. Finally, you have a summary of the statistics, like the chi-square of your fit, the reduced chi-square, and some of the some test criterion that if you want to test multiple model and you don't know which one is the one you should use, then that's something you can look at. There are modules to, so for example, if you want to run a grid search, like you are not sure for some parameters, or you want to see your chi-square surface for some parameters, this can be run, for example, this method here. You can define a grid, for example, in step 1, where you vary the exchange rate, the population, and the delta omega between, for the exchange rate 100 and 600. You use 10 points on the grid, and I use a logarithmic grid here because it's more convenient for exchange rate. Same for population, but a linear grid between 0 and 10 for the chemical shift difference. And the way this would run is basically exactly the same as before, except that now you have the grid that is calculated. So ChemEx tries to optimize all the calculations. So if you have parameters that are independent of each other, it's not really necessary to make a grid of those two parameters, or it could create subgrids. For example, delta omega of residue 12 is independent of delta omega of residue 13. So in that specific case, instead of having 10 power of 3, if I'm correct, points to calculate, it would just run 10 grids of 10 power of 2, which is much quicker to run. And I have another grid on delta omega in another step, so it does everything. And the same way profiles are plotted in PDF files, the grids will also be available either in text file or in the PDF files. Okay, so the rest of the calculation is just run. I can show you maybe one of the grids. For example, here you have grids that are summarized in 2D. So the color is a chi-square value, and that's between chi-exchange and delta omega in that case. You have, for all the delta omega that were included, you also have between the exchange rate and the population. Here you see a nice correlation actually between the two, but not too bad. And you see the grid, logarithmic grid is also visible this way. They are also summarized in 1D. So in step 3, I did grids on delta omega. Only delta omega is gridded, and you see the variation of chi-square as a function of delta omega. When it's flat, it's 0. That's pretty clear. And when it's not flat, you see a nice minimum here. So it's just a way of visualizing. So this is for CEST experiment, and exactly what I discussed now can be applied directly for CEST experiments. So I have one example for N15 CEST experiments on that folding reaction of a small FF domain. Basically, that's a folding reaction that is barely, you can't really study it with CPMG because that's too slow. And the dispersions are very small. But with CEST, you see nice minor dips corresponding to the excited states, and in this specific case, at the unfolded state. And I can show to you very quickly, but the structure of the directory is the same. You have the experiment files describing the module in ChemEx that is used, the carrier, frequency, and so on. And all the profiles are here, the data, and so on. It's the same, exactly the same. The parameters that are defined here are more or less the same. You have the chemical shifts, population, exchange rates, and that's enough. So if I'm going to CEST, the command I'm going to run is just this one here. I don't have multiple steps here, so I don't even have a method file. It's just parameters for initial parameters. And all the parameters are fitted all together here. And you obtained your CEST profiled in those files. So here, so you see the position of the minor dip, and that's it. So after you analyze those data for this system, we can see a very nice correlation between the position of the chemical shift of the excited state and the unfolded state. So it's most likely an unfolded state. And actually now there's a very nice study by Pramod Valurupalli and Whiskey that showed that you have actually other exchange processes in this minor state. And the kinetic model is actually more complicated. But if you record additional CEST experiment with different B1 field, then you can characterize it. It's actually 4 states, so it's quite complicated, but it's a very beautiful study that was published by Pramod in PNAS last year, I think. And Imanshu also asked me if I could show an example where I'm combining CEST and CPMG experiments. And there's one example where actually we were not studying especially like a conformationally excited state, but the interaction between an intrinsically disordered protein and a globular domain of a larger protein. And those can be quite challenging for a few reasons. in general, when you have this kind of interaction, the complex form, you have a part of the intrinsically disordered protein that would start to partially fold, will become more rigid. And this is a part that will probably be characterized easily by crystallography. Sometimes they even like cut the flexible part, but the flexible part won't be visible. And you have the opposite problem when you run an MR experiments, where the flexible part would give like very intense signal, but the one that is folded would behave as the larger object, and the relaxation properties are not that favorable, and most of the time you would lose that signal and you won't be able to see it. But if you do the same trick as I showed to you for the small peptide, you have the intrinsically disordered domain, you add a little bit of the partner, then the complex form, the bond form becomes a sparsely populated form that can be studied with CPMG and CEST. And on that system, that was the interaction of Artemis with DNA binding domain of the ligase 4. It's not all Artemis, actually, it's just a segment here. We collected multiple CPMG experiments with different concentration of the partner, from 3% to 10%, if I remember correctly, but also CEST experiments on one concentration. And all those data were run on the same spin system, that was amide nitrogen, so can be fit all together. So I'm going to show you, actually I can show you things here. You have this two-step binding, and now instead of two experiments, I have a series of experiments. So all the CPMG with 3%, 6%, 10%, and so on. Because the kinetic model now is a binding model, you need the protein concentration and the ligand concentration, at least the amount you add in your sample, for each experiment, and for the CEST experiment as well. And it's actually extremely easy to run the analysis on all the experiments together in a joint way. You can, I'm just showing to you the command line. This translates, basically there's a lot of experiments, so I'm using that star, it's just a shortcut to say that everything that ends with .toml will be included. And that's basically all those experiment files. And by doing so, you just add experiment files, and the data that correspond to the experiment files will be included in the fit. And here I'm just like running it with the first step on the interaction side. So it's 91 profiles. The same trick as previously, I just feed PB, it's not PB, it's K on here for those profiles. And then in the second step, I apply the same, I fix K on and I fit the rest. And so the calculation here, because there's many more data points, it takes a little bit more time. So I'm not going to wait for the end of it. I already run it, so let me get it. Can show you the results directly. So, for example, if you want to see the chemical shift difference between the two states, so that would be delta omega AB. There's a small command that can help you do that. So you see the sequence. You see that this is the interaction site where you have the chemical shift differences. But in that specific case, because we were titrating, we were able to also extract the R2 of the bond form. And in that case, that was actually pretty interesting. I can try to show it to you. Oops, made a mistake. Okay. I just want the R2. See, you see the R2 of the free form of the IDP. It's very small. That's what you would expect, and the R2 of the bond form. And it's actually pretty interesting in that case because those resonances, we don't see them in the experiment. So those values only comes from the CEST and the CPMG experiments. If you see the site where you have like chemical shift differences, it's a bit shorter than the segment where you have increased R2. So for this system, we haven't gone further than that, but we can suspect that there's some transient interaction that are extending outside the main, the core of the interaction, and that may lead to increased R2 values. Okay. And so from step one, for example, you would have, so in that case, we had the KD from different experiments. So we fixed the KD. Sorry, it's a bit. So the KD was fixed to this value, so it's micromolar, and K-off was varied. And the results are given here. So you have that value for the K-off. And the fit for delta omega. For CEST, you have R1 that is fitted. For CEST and CPMG, the R2, but the same R2. Actually, all the parameters are the same for the two set of experiments. So that's where you can really exploit the complementary between the two set of experiments. Okay. With that, I should, of course, acknowledge all the contributors of ChemEx. There are many. Of course, I'm the main driver behind the program, but I receive contributions from many people. It's been initiated in Lewis-Kay's lab, and many of the experiments that are implemented in ChemEx are the ones that are developed and used in Lewis-Kay's lab. More recently, I got help from Terran UN. Alexander Hansen has been a strong contributor from the very beginning. But not only people from Lewis-Kay's lab have contributed. As this is an open source project and it's available on GitHub, anyone can come and contribute. So I've been very happy to interact with Renio 10 that contributed a lot to the program. I must say also that at the very beginning, I got a lot of the inspiration for ChemEx from a program called Katia that was written by Fleming Hansen for CPMG experiments and two-state exchange model that was using the same kind of formalism. But that was just for CPMG experiments, and I wanted to do some more, so that's also why. And also like interaction with other people's when I was collaborating that suggested new ideas. So that's very important sometimes to get the program to work better. With that, I also would like to thank you for listening to that presentation. And I'll be very happy to answer questions or to go into more details in some aspect of those calculations I've been showing, if you want to,
yeah.
Or anything. Yeah.
Yeah, thanks a lot, Guillaume, for your lucid presentation. It's really nice, and now I can imagine fitting CEST and CPMG together. So that's really cool. Maybe if you have any question for the audience, you can raise your hand and directly interact. In the meantime, I will start with one. Suppose that we have a protein and there are different time scale of motion, and it is in microsecond, millisecond regime. So it is going from something like, if we try to scan, like fitting individual residues, we have motion going from 100 microsecond up to 500 microsecond. then you showed this group fitting, fixing K exchange for a global fit. So could you comment on this, how to get this global parameter? We can select very well like similar behaving residues and we can fix with that value, or what is your take on this?
So it really depends. So
yeah.
If your exchange is really like two states exchange, all the residues will see the same exchange rate and population. And in many cases, if you take a few good profiles you have, the minimization is pretty straightforward and the starting value is not that important. And you very easily succeed in getting those parameters out. If the exchange is more complex, then it requires a more complex model. That starts to be a little bit more work because the initial values can be important. So you may have like different clusters of residues affected by different exchange rates. If you're lucky, you can distinguish them and feed them separately. It's been done in a few systems that I know of. If it's really like, for example, a folding reaction with an intermediate, and you see the unfolded state, the intermediates, and the folded state is a major state, then you really have a three-state model. So then you can start trying to do some maybe grid search or this kind of approaches. More recently, I haven't mentioned it, but there are a third way of analyzing those data. So there was analytical formula, the numerical integration I shown. More recently with machine learning also, you can train models for specific experiments and you can train them for a different kind of kinetic model. So Fleming and Sen recently published something on that. And that's a way where you don't really have to fit. It's like the algorithm recognize the profile as being like three-state with those parameters. And it comes out pretty, so it's actually a very nice approach that can have a lot development in the future for this kind of application. One way of dealing with when you have a bit of uncertainty, you can start with two states on a residue specific basis. So I can show briefly to you how that could work very easily with one line. So let's go back to the CEST dataset, for example. The command line I've been running was this one. So if I run the exact same one, but I want residue specific, so I change the output directory, I can select from the command line the kinetic model I want. And there's one that is two states, residue specific, maybe it's underscore. The website would tell me right away. And if I run this one, then I have a residue specific population and exchange rate. And that's what will come out also. So if all your residues will provide you with population and exchange rates that is more or less the same, it's a very strong argument to say like, okay, I only have one process. All of them are seeing the same. So this is something you can check by looking at the resulting fitted parameters. And you see that now the exchange rate is residue specific. And it's mostly around 55 for most of those residues. The population is also pretty stable around 1.5. And if you run the fit with only one population and one exchange rate, so it's a simpler model, the chi-square will not increase. So there's no reason to go to more complex model. As I said, there are more complex models available in ChemEx, for example. Not sure it's available in the version right now, but there's a model for binding through an intermediate that could be like conformational selection or induced fits. Then you would have like two pathways. These kind of things, you have more parameters, it might be more difficult. So if your system allows to define those parameters, then there's a solution and you ultimately be able to find it with maybe some grid search or smart.
Yeah, thanks a lot. Sarah, please go ahead with your question.
Hi, thank you. Thank you so much for the presentation. My questions are a little bit more on the practical side, and maybe they are a little bit dumb. So for the input files in the third column for the CPMG, you require the uncertainty, and you were also referring to it as error. I usually get my pick list out of POCI or CCPN, or Sparky or CCPN. But I don't know exactly how do you calculate this error, because usually for other relaxation dispersion analysis that I have done, the information that is fed in is either the signal to noise or the signal to noise per peak or for the entire spectrum, and from that is calculated the error using Monte Carlo. So I don't know exactly how to calculate it and how to feed it into the the input files.
Okay, so a few points here. You can provide it, but if you have duplicate points in your profiles, and that's very, for CPMG, it's something that's should probably do. Yeah. And for CEST, you don't really need them because you have the scatter on the baseline. You can estimate the noise directly in ChemEx. There are two algorithms that are designed for these specific things. This is this line here, scatter, or for CPMG, if you have duplicate points, you would use like duplicates, or that can be files, and then to take the value in the file directly. So for CPMG, the duplicate is something called the pooled standard deviation that's taken from the duplicates. For CEST, it's a little bit more fancy algorithm, but basically it looks at the scatter on part of the profile. If now you want to estimate them, the way I'm doing it, so we are pretty lucky for those experiments that we are dependent on the variation, not the absolute value of the intensity. That means that you can work with either the peak volume or the peak height. And a very easy estimate for the peak height uncertainty is to take the standard deviation of the noise in a region of your spectra where there's no signal. So if you take that value and you put it directly for that uncertainty column, and on the intensity you put the peak height, you would be perfectly fine in most cases.
Okay, so that would be kind of the peak list that, for example, you get when from Sparky when you select the signal to noise column, because I think that this is the value that is calculated.
Yeah, in Sparky, I believe there's one place where there's an estimate of the noise, so maybe that estimate is good enough to take.
Yeah, okay.
And the peak height would be used in that case, not the peak volume. That would be
I see.
Different, yeah.
Yeah, okay. Thank you so much.
Yeah. Rasmus, please go ahead.
Thank you also from my side. That was really beautiful. Yeah. I have a few questions and I'm not sure where to start. I guess what's maybe most or easiest or I don't know, the best thing to ask first is, so if you have multiple steps of some confirmation exchange process in some sample, then and you do this, for example, 3-step or 4-step fitting, is that, or what kinds of data would you take for this? Because I can imagine that it's easily underdetermined if you just work with only 15 ENS profiles, for example. Or is it like, do you get all of the data even just from that because you have multiple dips, or do you take C alpha CEST into account, or I don't know what?
So the question is, if you have multiple steps and if you don't fit enough data to get the parameters well defined, so yeah, that requires some knowledge about the experiments. And the basic rules are, when you run CPMG experiments, it's very nice to have at least two B0 field data sets. The reason is very simple. The chemical shift difference in ppm is the same in the two sets. But in radian per second, it's different. So you're not exactly in the same exchange regime. So when you mix the two, you're already constraining your system because the population and the exchange rate are the same for the two data sets, but chemical shift difference is slightly different in radian per second. It's proportional to B0 basically. So this is the usual way of doing things. If you only have one B0 field data set, then you have to be a bit cautious. The other thing is for CPMG, it's better if you can fit multiple profiles together, because it's very easy to have like exchange rates, population, and chemical shift differences to be covariating, like really not defined very well. But by adding a few profiles, you start like constraining your system very much, because population and exchange rates are the same for all those profiles. So if your system is a two-state system, then a few profiles, maybe 5, most of the times would be enough. Then you just take the one that looks, shows a nice dispersion. Of course, the ones that are flats, they contain not much information about the exchange. They can be useful. I mean, when you have the exchange rate and population, you can fit them and you get a delta omega of 0, which is some kind of information. Sometimes you know, okay, that part of the protein is not affected by the exchange. So good. For CEST experiment, the rule is slightly different. What is important is to have different B1 field. So you can run on the same spectrometer, the same experiments with different B1 fields. And what's applied for B0 field is almost applicable to B1 field. So when you vary B0, the higher B0 is, the slower the exchange regime, because you're increasing the chemical shift difference, so you're pushing your systems towards a slow exchange regime. And you have more or less the same kind of effect with B1. When the B1 starts to be higher than the exchange rate, you achieve some kind of slow exchange regime. But before that, the dip would vary with B1, the height of the dip would vary with B1. So if you have multiple B1, then you start constraining your systems and you start being able to extract, for example, the R2 of the excited state, having it be, we've seen that residue-specific CEST experiment, like the examples I was showing, is only 2 B1 fields, and I already have the same population and exchange rate for each residue independently from each other. So it starts to be already pretty robust. So look at the error bars, run maybe Monte Carlo simulations or bootstrap analysis to see if there are correlations between parameters and if you can rely on the one that are obtained from the fit. But if all those lights are green, then you're good to go. I don't know if there was more to your questions.
Yeah, no, that's good. Yeah, various aspects of which I hadn't thought really. Or some of them, yes, I know, but others I didn't. And so is there a possibility also of adding other perturbations to your sample? Like, I mean, not necessarily the NMR data. You mentioned like the B1 and the B0, but for example, temperature or external conditions, I don't know, the population, like once you had this, like the equilibria between a bound state and an apo state, then the concentration of the second sample was something that you would have to kind of fine tune to test the methodology. But if you have a real like biological question, could you play with the concentration and also kind of involve this into the Monte Carlo or into the data fitting, like a principal component?
Okay, those are fixed parameters. Like all the, I'm just like, okay, it's not available yet, but there will be a website, and I have the list of the kinetic models that are actually implemented. It's, as I said, modular. So if needs be, we can add more than the one that are here. But you have, for example, like models that depend on the temperature. So in your experiment files, you have to provide the temperature, and that would be a fixed parameter. So you have to get a good measurement of it. And for the binding experiments, also you have the protein concentration and ligand concentrations, and they are to be fixed parameters during the fit. If you want to see what would happen, like you have exchange parameters, like binding parameters for a system, and you want to see what would happen if you have a concentration of that value and for the protein or that ligand, there's a small simulate module that does simulation. So you can create like fake experiments and run ChemEx to calculate the profile that would be obtained. Sometimes useful to when you want to prepare an experiment to see what parameter would be the best. If you have a small idea of the exchange parameters, that's a good way of doing it.
Nice. Maybe one last question that before I let Benedict ask his question. So I'm not an expert in CEST, but I know that so if you have proton CEST, then there's always an NOE contribution. Is that something that is somehow exploitable in the sense of, for example, when you want to know something about what the excited state or the bound state, for example, looks like? Can you derive, for example, for this minor conformation, some kind of additional like distance restraints or something from this?
That would be really nice. It hasn't been done, and probably because first, the NOE deep in the CEST profile, the proton CEST profile, we have seen as annoyance than something of information. And they all come from the major state. So the proximity in the major state would give that small dip that comes from the NOE. There could be an effect when it's bound, for example, or in the excited state, but that would be probably too minor to be seen in the profile.
It's just not long enough for the NOE to build up, right?
Yeah, and the one we see are the one have the same size of exchange dips. So, and they comes from the major state that is populated like 95%.
Unless you have something like that doesn't have any NOE contributions, like for an IDP, and you don't have any I don't know if they probably don't have so much NOE, but then, for example, when it folds into an alpha helix.
That's a very interesting point. I mean, that's something maybe to simulate, actually. The way it was seen previously that those dips can be like, they can be many on your profile. So we worked hard on trying to find methods to get rid of them. I tried one experiment like a few years ago, and more recently there's a very elegant solution that was found by Terran Yuan and Lewis K, where they were recording some kind of TROSY CEST and anti-TROSY CEST, and when you do the subtraction of those two experiments, the NOE contributions vanishes, and you only have exchange contributions. So that was a very nice things, because otherwise it can be like, I don't know, 5 or 6 contributions from proximal protons. And that's very difficult to analyze. So.
Yeah, right. I heard about this somewhere, the TROSY and anti-TROSY thing.
That's pretty elegant, because the NOE is not dependent on the state of the nitrogen. So it does not change in the TROSY or anti-TROSY profile, and you can subtract the two. Have this kind of anti-phase profile. And,
cool. Yeah, thank you so much.
Very nice. Yeah. Benedict, you may go ahead. You are muted.
Yeah.
Sorry. Hi, thank you for the nice talk. I just have 2 questions, practical questions. One is just regarding, I'm not sure if I understood it correctly. So if you wanted ChemEx estimates the intensity error for for example, if you measure 2 CEST experiments or 2 not CEST CPMG at 2 CPMG frequencies twice, then you don't have to put any intensity error to your input files, right? Or was it like that?
Okay, it could be this way. It's not right now. Right now, you have to provide that column because it's expected by the program. Mm-hmm. Even though it can be overwritten by the methods I discussed previously, if you have duplicates or if you look at the scheduler. So you can put a fake value for the moment. Maybe that's a good point, that if it does not exist, then it tries to calculate it directly from the profile. It's a good suggestion, actually. Just,
okay.
Yeah, I must say, those kind of programs, the first time they are written, it's really to solve the specific problems you have on your daily basis, and then you share it, and there are more applications, and people are using it in very different ways, and that's really where you learn to make it better. So it's been evolving quite a lot over the last few years. I think it reached a pretty stable state now, and but small additions like that can be good additions. Oh, by the way, just to mention what I'm been showing today is the very last version. I will really publish that version in the coming days with hopefully this website. So don't be surprised if it's not exactly what I've shown that you would get if you download it. And the most recent version, the very close version you will find is the one on GitHub directly. There are other ways of installing it. I mentioned with Anaconda, if you have the Conda stuff and you're familiar with that, it's very handy, but that's not the last version. It will be soon, but not now. And with PEEP also, with Python, where I used to use that PEEP stuff, and it's also a previous version. But if you want to install that one, then just have one command line to copy and paste. If you have Python 3.9 installed in an environment or whatever. If you have any difficulty with that, just get in touch with me, or even if you have sometimes now I can also show the demonstration of how it's you can install it. And all the experiments also will be like this is the list of all the experiments that are implemented in ChemEx. So you have a series of CEST experiments. More recently, I developed NT-CEST for faster experiments. It's also there. For each of them, you have that configuration file that is the template. You can copy and paste and change the values as you wish. Yeah, hopefully all of that will be on the web in maybe before next week. I don't know, very soon.
Okay, thank you. And then I have a second small question regarding the input files. Is there any program which by default generates the input files like that, or do you always have to somehow convert it with some small Python script?
The intensity profiles, it's basically the output files of the program FUDA by Fleming Hansen, or the program I wrote, PeakFit, would also give you the exact same format. For ChemEx, this is basically why I put those experiment files. You can just click here, then you copied it, and if you create I don't know, a new file, you can just paste it and then change the value according to the experiment you're trying to fit. For the methods, files and so on, they are changing too much for me to have maybe I can have a few templates, but then I would direct the person to look at the examples. I can, okay. The examples, so it's, for example, in if you want to run CEST experiment, you have an example of a fit here. So the example of a parameter file. Again, you can also copy and paste, and but you have some editing to do. There's some people, I think it's called NMR Online. I haven't seen the final product. It's still in beta. I think it's been advertised at Euromar, but I was not there.
NMR box or?
No, it's not an NMR box. So NMR box includes ChemEx, but an old version right now. It will be corrected, I guess, soon. Mm-hmm. No, it's called NMR Online, but they have a beta. It will be purely online, I think. And they told me that they will implement ChemEx with a graphical user interface. So I haven't seen it, but maybe it's coming soon via NMR Online. For the time being, yeah, you have to edit a few text files.
Okay, thanks. Thank you.
Maybe we can take some more, maybe 2 questions. Sarah, you may go ahead.
Thank you. Sorry. Yeah, so in NESI, another program to analyze CPMG data, something that you get as a result is a comparison or a decision if you are in the fast exchange or slow exchange. And this is basically the main result that the program is giving to you. Do you get something similar from the ChemEx, or is something that you have to decide on your own on the basis of the output files?
So it's not in ChemEx, and there's a reason for that. I will go back to this table here. So what you're referring to is to fast exchange or slow exchange or intermediate exchange. You have an analytical formula for each of them. So it's basically those approximative solutions I was talking about.
Okay.
ChemEx makes no approximation. What would happen though, for example, the fast exchange model for CPMG, you don't fit the population and the chemical shift differences. You fit a parameter called phi most of the time that would embed those 2 parameters together. It's something like P, population of the excited states times the delta omega, the chemical shift squared, and that fit is reliable. So you have to see that, okay, population and exchange rate, if it's in a fast exchange regime, would be correlated, and you won't be able to get the 2 accurately. So in that specific case, the most obvious things to do is to fix one of the 2. Then at the end, you can recalculate that phi parameter, if that's the one you're interested in, by taking the population and the chemical shift differences that gets out of it. But the Monte Carlo simulation would directly provide you that information. Okay, I'm in fast exchange and those parameters are not well defined because they are covariating. So that's why you don't have that idea of fast or slow exchange in ChemEx.
Okay.
Basically, the formalism that is used works for both.
Okay. Yeah. Okay, thank you. It wasn't clear for me at the beginning. I thought that, or I didn't correlate that it was either or. I just thought like there are pros and cons, and maybe it uses both, or yeah.
Can you repeat your questions, because I got the video stopped for a few seconds.
No, it was not a question. It was just that it wasn't clear for me at the beginning that, or from the presentation, that is only using the numerical integration. But yeah, now it's pretty clear.
Yeah,
thank you so much.
Yeah, I must say it's modular enough to be able to also add modules that would be just analytical formula. But the advantage of that for now is not obvious to me, because simple models they are fast enough to be calculated with the numerical integration, which is normally more accurate.
Yeah, I see. Yeah, thank you.
Yeah, it's very interactive. Another question from Rasmus, maybe.
Yeah, just something that I also wanted to ask you. You had this example of this IDP that somehow samples the surface and then interacts in a certain region, and also has a certain region of interaction in its own, so very much localized to some stretch in the N-terminal region. And then you saw that whereas you had the chemical shift perturbations pretty much restrained or restricted to that region, the R2 increase would extend a little bit beyond that zone. And my gut feeling would have been that since you have something that binds, and then of course the rest is floppy, but it is kind of just becoming more and more floppy the more you go away from the binding region, that basically your R2s would finally get to the normal free apo form R2. But when you're pretty close, then your order parameter is still not at 0, and you still have certain restrictions, and that even without doing the sampling, you would already get some kind of R2 increase. What's your take on it?
No, I think that's a very good observation. You have steric contacts also that can happen. The thing in that specific case is it's not symmetrical over the core of the interaction. It's really more, it can be a length effect, but I don't know if that would behave exactly like that, actually, if it was just the length, because you've seen the core of the interaction is really on the N-terminal parts. I'm talking about it, maybe I should try to find it. Yeah, but it was things I was showing previously. Okay, I don't have it anymore. Yeah, but the fact it's not symmetrical makes me think that there's something more. What exactly it is, we are not so sure. When we look at the protein, it's pretty clear that there are a positive patch on that side, and a negative residues on the IDP side. So, but it can be different. I don't know exactly.
The same kind, maybe.
Yeah.
I guess the various mechanisms come into play at the same time. The question is how to disentangle this. But I mean, the sampling of the surface is makes sense too.
Yeah, we haven't investigated that system further because we were more interested in the full size domain. But I must say that this kind of study with an IDP and an interaction with a globular domain where you can run CEST and CPMG, it's one example of, maybe not many, but really a few already. So I got some collaboration with Malene Rinkyobin Jensen, and she's got a few examples. On our side, we have additional examples where it's exactly in the right time scale for doing this kind of experiments. And it's very difficult to see the bond state in the normal spectrum. So that's very adapt to this kind of study, actually.
I also have an application in mind already.
Yeah.
For one of our samples. Yeah, that's cool. I think it's yeah, exactly. It goes more into this direction of DEST rather than CEST, or I mean, yeah, like you have a bound state and you kind of observe it. It's like a little bit like the ligand observed effects for like drug discovery studies. So yeah, I think there's a good range of applications, I would imagine.
Yeah, definitely. They are pretty simple experiments to analyze, because visually it's nice to have something that looks like a spectrum with test experiments.
Yeah, I mean, and having excited state shifts from just a single field is also cool. Yeah.
And I haven't mentioned things, but CEST experiments are not very sensitive to the presence of scalar coupling, so it's very easy to probe multiple carbon spins, for example, in a uniformly C13 labeled sample, which is a real issue for CPMG experiments, for example, for the aliphatic carbons. So it's very forgiving experiment. So as soon as we need a chemical shift and we can probe on one site, usually we can probe other sites as well. So it's good.
And is there any way of like overcoming the time scale boundaries? Because usually we're restricted to like pretty slow motion, which is kind of a drawback. But is like for CPMG, you have the, I don't know, the high power CPM, no, high power R1 row versions. Is there any idea that you, I mean, I have not been thinking about it.
For CPMG, I think there's a, is it Dungan Lee? I think
that is extreme
CPMG.
Exactly. But for CEST, I mean?
For CEST, there's a paper by Hashim Al-Hashimi where he pushed CEST to a faster exchange regime and got pretty nice results. By faster, it could be a couple of thousands per second, I think. In that regime, you don't really see a nice minor dip like in when it's a slower regime. So that's probably why we didn't really consider CEST as effective in that regime. But the profile still have some information about the exchange, and it was seems to be pretty powerful also for a faster exchange regime. As I said previously, there's a correlation between the exchange rate and the B1 field you should pick. If it's fast, you should try to pick a B1 field that is more or less on the same regime, same rate. So for example, if it's 1,000 per second, you should find a B1 field in radian per second. So it's not, you have to make the conversion from hertz to radian per second. That is about 1,000, and maybe one that is a bit lower. Otherwise, there would be some correlation probably between the relaxation rate of the sparsely populated state and the exchange rate, for example, or some other parameters.
Cool.
And yeah, so that's one of the idea. The other idea for CPM, so you have exchange-induced shifts that don't behave exactly as exchange-induced relaxation contribution. And if you combine CPMG and those exchange-induced shifts, like if you look at shift in an HSQC and an HMQC experiment, you can basically break that correlation between the population and the chemical shift difference. And then you can get all those parameters well defined for a few thousand per second, even 10,000 per second. There were also ideas by Pramod Valurupalli recently where he was looking at multiple quantum coherences, like triple quantum coherence would have a delta omega that is 3 times larger. So that pushes the systems toward a slower exchange regime, but also the chemical shift is pushed also in that slower exchange regime much more. So you have also larger shift, and you can combine those two and get to, I think, 10,000 per second and still get all the parameters well defined, because that's really the issue with fast exchange. So if you do extreme CPMG, you would have some nice dispersion, but from those dispersion, it's hard to get population and chemical shift differences altogether.
But so yeah, I know the triple coherence CPMG experiments. But CEST, that's obviously a nice idea to
yeah, it can be. So mostly they are on protons. There are some quadruple quantum coherences exploited also by Chris Woodby is talking about that these days.
Actually, I heard a talk by Daniel Heusinger, and they were using contact shifts to increase the bandwidth of like the kind of the shift differences between excited state. I don't know what the excited state was. It was some kind of ground state and excited state in CPMG experiments. So you would have also increased sensitivity, and maybe that would also work for CEST. Probably.
It's for CEST, it's a dream. And actually in imaging, you have paracest. It's exactly that. So you can apply the same, and recently Dorothy Kern also used pseudo-contact shifts in the excited states. She measured them with the CPMG, but it would work very nicely with CEST.
Exactly. That's the same talk. Like Daniel Heusinger is the person that made
the tag. Yeah,
the tags.
Exactly. Yeah. And the big issue is the tags. So in the paper, I think the structure was obtained from a metal binding protein. So they didn't have to put a tag in it. But there are issues with many tags, and I think they kind of solved that issue these days. It's like the motion of the tags can create dispersion on top of the conformational exchange dispersion you are interested in.
Yeah, that's true.
And that can be an issue. It might not be an issue for CES, because if the exchange of the tag is fast, then it's just an average value we would obtain, but not necessarily something that would perturb too much the profile.
Mm-hmm.
So on that side, I would say also CES is probably more resilient. The fact that you get the chemical shift rate out of the profile is a big advantage, I think. And that's basically one of the most accurate parameters you can obtain from CES experiments. So that would work perfectly for pseudo-contact shifts.
Exactly.
Yeah,
unfortunately, I have to leave soon. We have a visitor over, but maybe I can ask you one last question if you mind.
No problem.
So you were also saying something about the Dante test that you were developing. So that is a way of just getting multiple information at the same time, or multiple spectra instead of a stack.
Okay, maybe a picture would make things clearer. I will just.
I know that there is this quick version, but I don't somehow derived from some work of Louis K. But I don't know exactly what they do and whether that's what you've been.
What version what version are you talking?
I don't know. I just know some paper that kind of gets the multiple, in a pseudo 3D, instead of using multiple 2D spectra one after the other, somehow combines this into a single spectrum and then gets this faster. But I don't remember. Maybe Zurich remembers. I guess we were talking about the paper at some point. I'm not sure if that is done to test, or that's something different.
Dante is slightly different, and it's mostly used for. Okay, I think. Okay, it's mostly used to speed up the acquisition by far, actually. So those are profiles that have been recorded on a gigahertz spectrometer, and you see there's many points that don't bring information because it's a sparse spectrum. You only have 2 resonances. And one way to compress that, the one I found, is to have multiple excitations. Dante is very simple to work with and would generate an excitation profile that looks like a comb here. So instead of having one frequency excited, then you have multiple ones. And when you do that, basically you can record only one period and you have all the information. The rest of it is just periodic, so you don't need to record it. The thing you may lose using DENT is the absolute position of the peak, but the profile behaves exactly as if you have folded peaks. So if the peak is on the edge outside of the irradiation window, then if you change the spectral weights between 2 experiments, it would move exactly as if it's folded in a normal spectrum. So actually you run CES usually using several B1 fields. So you can run that Dante scheme at one B1 field with 1 spectral width. You increase the B1 field, you increase a little bit the spectral width, and you don't lose any information at all. But instead of recording all the points, you record a quarter of the points. And if you use very small B1 field, then the line width of the dip is smaller. You need more points. So that was an issue before, but with the Dante, you just use the same number of points. You can even decrease the spectral width because the line width is smaller. And then it's become manageable to run very small B1 field experiments. So that's the idea behind the Dante scheme. That's cool. So that's actually actual data that were recorded normally or with the Dante.
Yeah, that's definitely something different then, but it's cool. Yeah.
Yeah, if I run an N15 CEST experiment these days, I always run with the Dante because even though if I want full spectral width, I just increase the spectral window and it works fine.
Yeah.
Equivalent, the CW irradiation of the Dante irradiation, the line of excitations, they have the same shape. They are the same.
Cool.
Yeah, very nice, Guillaume. Really very interesting and lots of questions. So we can see the involvement of the group to the topic. So it was very interesting. So we are very happy to have you today, and actually we enjoyed thoroughly. So I am not able to see any other questions. So if not, then we are very close to the end. So we all can thank Guillaume for such a wonderful talk and very live demo and demonstration and actually question answers session. So thanks a lot for joining us today. And it was really nice.
Thank you.
Yeah.
Yes.
Thank you to you.
It was very nice meeting you.
Yeah, nice meeting you too, guys.
Bye.
Bye.
Hope
Bye.
to see you soon. Yeah. Bye.
Bye.
Okay. Thank you.